Happy Monday, everyone! It's a new week for me in my thesis year, my final year of bioscience engineering. And on Mondays I follow lectures. I have lectures, but I don't have to go to the campus because it's pretty far for me, about 2 hours one way. So I follow them online. And I'm also busy with some primer design for doing a PCR, so I can continue some new experiments for the coming weeks, because I'm almost done with my first large experiment. Watch my previous video for that. Quick training session at my home gym. And of course keeping the frogs unstressed. Let's go, Willem! Alright, and my colonies have grown. Watch the previous vlog of last week to know what I'm talking about. And today I'm going to do a colony PCR. It's going to take the whole day because I have quite a lot of samples. And if you don't know what a PCR is, it's like you might have heard about it from COVID. It's the same. PCR is basically multiplying a very specific small piece of DNA in billions of copies in a reaction, so you can actually detect the DNA. That's how you can know if there was any DNA of that specific copy to begin with in your sample. And the colony PCR is doing that on colonies of bacteria. So I'm going to check these bacteria colonies on a specific piece of DNA. Which DNA? Well, the plasmid I made last week, because they should contain them. Let's do it.
Here I'm picking every colony separately and mixing the cells in a drop of water. The cells are then microwaved in order to break them open and release the DNA in the solution.
Alright, I'm in the clean room, and here I'm going to prepare the master mix for the PCR reaction. So I already prepared the DNA from the colonies in the microwave, in the water just. And now I just have to prepare all the ingredients, let's say, to make the PCR reaction happen. I first had to calculate how much of each ingredient. These are the ingredients, by the way, if you're wondering, I have to put in each tube. So I'm basically making 2 mixtures.
The final step to prepare the PCR is to add the master mix to the DNA water solution.
Alright. The PCR is running, so now if there were any plasmids in my colonies, then they will multiply billions of times in the PCR reaction, and then I can actually visualize them when there are many copies. So yeah, hopefully that's the case. And now I'm designing some primers for other PCR reactions. Primers are the factor that makes it specific that only one piece of DNA can multiply in many copies. Everything is ready to put on gel. So the PCR reaction, it took about 2 hours. It's ready. So hopefully the specific fragments from the plasmid are multiplied billions of times and are now present in my DNA. I have already prepared. I have 57 samples, 8 colonies of each of the 2 plasmids, so that's 16. I'm going to check 3 fragments of each, so 16 times 3, and then the positive and negative controls included. We come to 57. So yeah, now I have to load all of these on the gel. I've prepared 2 gels. I can put it in this one and this one. The first gel is loaded with 2 times 18 samples. Let's do the second one. And these things are the molds for the little wells I put the gel in. So when I pour the gel, it's still a liquid and then it solidifies. Alright, this shell is also loaded. Just to recapitulate, if you haven't watched the previous video, this is a gel electrophoresis. So in every little well, there is one specific fragment of DNA, a lot of copies of it, so billions of copies of one fragment of DNA. And I apply an electrical current on it. DNA is negatively charged, so it goes to the anode. And the gel for the DNA, because it's a very small molecule, is basically a big mesh, and it tumbles through it, hits things, it moves. And the bigger the piece of DNA, the more resistance it has through the gel. So if it's a big piece of DNA, it goes very slowly. If it's a very small piece of DNA, it goes a lot faster. Based on that, you can determine the size of the fragment of DNA in your sample. Again, 40 minutes on 120 volts. 1 minute left. Let's put it under UV light, the gel, to visualize the DNA. Because the gel red is in the gel and it binds the DNA. Moment of truth. This is what I've been working on the whole day. Okay. That looks pretty good. Alright, I have some positive results, so I'm very happy. I've officially made new plasmids. We're still going to send them in for sequencing, but normally it should be okay. And this we just throw away. Took a picture of it, so I don't need it anymore. It was just to check if my colonies contain the plasmid or not. Awesome, very happy. Okay, in here I just inoculated the colonies that were positive, so I can grow them. Tuesday evening, catching up on some classes from laboratory animal science. It's 9 p.m. now, and to finalize my work for today, I'm working on the frog video. I need to make a DNA vector to transform the embryos, and I'm still reading on which method I'm actually going to use, CRISPR-Cas or a meganuclease. I have to go to campus in Ghent, so I have to travel for 2 hours just to follow some classes. So, when we're looking at the it's 9 p.m. Let's go home. It was a long day. They have grown. Let's do another plasmid extraction.
Here I'm performing the next steps in the plasmid extraction protocol, basically ending up with a pure solution of the plasmids the bacteria cells produced.
Alright, so this is my purified plasmid, and now I will measure the concentration of DNA. I'm gonna do that with the nanodrop one, very easy. Now I'm going to transform these bacteria, it's just E. coli BL21, with the plasmids I just purified. So it means I'm just going to put these plasmids inside the bacteria. Then they can actually start to produce them. And I'm going to do that with a heat shock. This basically means that you give the bacteria a heat shock, so go from very cold to very hot very sudden. Then the membrane of the bacteria gets small holes, and through these holes the plasmid, the little piece of circular DNA, can enter the bacteria. So let's do it.
Here the actual heat shock is performed for 40 seconds at 42 degrees Celsius. This is a step where the plasmid DNA will enter the cells. Here I'm adding special media the E. coli cells need to recover from the heat shock. I will incubate them for 1 hour in this media.
So now I'm preparing some plates with LB agar. I'm also going to put in some antibiotics because after an hour that the bacteria have been incubating, I'm going to put them on these plates and I have to select for the plasmids that I just transfected the bacteria with.
Thus, here I'm making plates to grow the transformed E. coli bacteria on. The final step is to plate the bacteria out on the agar plates I just made.
I did the final thing I had to do today in the lab. I made a plate for Sanger sequencing. So the plasmid we did the transformation with in the E. coli today. We're going to sequence the actual plasmid. So sequencing means just figuring out the exact genetic code to check if it's perfect or not. We're going to send this to another faculty nearby, but for now I'm going to put it in the freezer. Then I'm done in the lab for today. It's now 9 p.m. Since I got home, I've still been working on something for my thesis here, and I guess I have about an hour left of work. Alright, and as you can see, my colonies have grown, all of them, so that's very good. And today I'm gonna do another colony PCR on these ones, just like Tuesday. So I'm not going to film too much, it's basically the same. So I'm going to figure out if the DNA I put in these cells yesterday, the plasmids, are in it or not via PCR. The gel electrophoresis I will have to do on next week Tuesday because I only have half a day today because I need to be somewhere in the afternoon. And again, Saturday is thesis writing day. Happy Sunday, and every other week I have to work in a restaurant in the kitchen. Would you look at that? I'm back home from work, it's about 6 p.m. and it's time to feed the frogs. By the way, I feed them almost every day, so it's not only on Sunday. And just as last week, Sunday evening is video editing day. The video you're seeing right now actually. Thanks for watching and I see you next time.